Usually what happens is we collect suggestions from CCMB folks. So whoever suggests them ends up introducing them.
Okay.
So now you know.
Start.
Soujanya, your mic is off, I think.
Okay. Good afternoon, everyone. Welcome to CCMB Biologue. CCMB Biologue is a student-led initiative where we take speaker suggestions from CCMB folks and we organize a talk every month. So far, we have had 16 such talks given by eminent scientists covering a range of topics from across the globe. The format of the talk is that the speaker usually gets 40 to 45 minutes, followed by a Q&A. The Zoom audience can pose their questions in the Q&A box or raise their hand, and the YouTube audience can pose their questions in the comment section. Today we have with us Dr. Ramakrishnan from NCBS, and I would like to invite Dr. Janvi Joshi to introduce the speaker.
Thanks, Soujanya. I'm very happy to introduce today's speaker, Professor Ramakrishnan from National Center for Biological Sciences in India. Uma is a leading molecular ecologist in India with a research focus on genetic data to understand biodiversity dynamics in Southeast Asia, especially among mammals and birds. Uma obtained her PhD in population genetics from University of California, San Diego. Then she continued her postdoctoral training at Stanford, and since then she has been a faculty at NCBS. She has received many awards, including Parker and Gentry Award, Outstanding Scientist Award from Department of Atomic Energy. She's also very passionate about science communication, and not only to the citizens, policymakers, conservationists, across students. She trains next generation students and scientists, and it's been an inspiration to many students and colleagues, including me. And today she is going to talk to us about how molecular data can inform tiger conservation. Over to you, Uma.
Thank you. Thanks, Janvi, and thanks all of you for coming. I do give a lot of talks, so I'm surprised when anyone shows up for any one of my talks. So I'm going to go ahead and share screen. Yeah. So while I'm going to talk primarily about tiger conservation genetics, I'll focus a little bit more on some recent work we've done on small and isolated populations. Can you hear me? Sound good? What happened? Sorry. Can I go ahead?
Yes.
Yeah, great. Yeah. So biodiversity is really important to all of us, and it's critical. Not just because it's beautiful, but it's really critical to human well-being. And the last several years have seen this very strong realization, apart from just thinking about the detrimental effects of climatic change, of global change, we realize that biodiversity loss is very strongly coupled to climatic change and vice versa. So the feedback, negative feedback between these two puts our planet and all of us in a very vulnerable position today, much more than we have ever been before. And when you look at this, even just in terms of, basic stats, which you might see in the BBC, about a third, of coral reefs, for example, are endangered. A fourth of all mammals are endangered and at risk of extinction. So this is not like, a small problem. It's a very large problem with a large proportion of species facing extinction, and even things like insects, many of which are seeming to be in massive declines. So as scientists, how do we address this issue, right? How do we think about extinction? People have worked on this for many, many years: ecologists, theoreticians, experimental scientists. And basically, the paradigm which we have is that small populations are more likely to go extinct. And you can see that here for this, this is data from butterfly. And you can see that, when the population size is very high, the probability of extinction is almost zero. But when the population size is low, the probability of extinction is quite high. And in fact, there seems to be like a threshold size of about 100, where, there seems to be a switch, and suddenly probabilities of extinction go down. At the same time, isolated populations are more likely to go extinct. So this is another, some more data from butterflies. And you can see that when you have a few isolated patches, the time to extinction is low. So they go extinct fast. But when you have many large or connected patches or populations, they don't go extinct as fast. So extinction then, so far as we have discussed, depends on population size and isolation and connection. But of course, extinction also depends on genetic variation. So basically, how does it depend on genetic variation? There are both direct and indirect effects. When you have small population sizes, individuals tend to be related, and mating between relatives results in inbreeding, which results in inbreeding depression, the loss of fitness due to expression of recessive deleterious alleles, and this causes extinction. On the other hand, stochastic processes are operating in small populations, and this one major stochastic process called drift, where this chance sampling of alleles can result in by chance fixation of non-good or non-advantageous alleles, and also push populations to extinction. And for example, here you see a really classic paper, Sacheri et al. This is around the time I started my PhD, so I was very influenced by this work, that when you have butterfly populations with a lower proportion of their loci being heterozygous, extinction probability is high. So what I'm going to talk to you today is mainly about tigers. But I argue, just as many of you work in the biomedical sciences and you think of, you use your systems to study something general, species like tigers are models to study extinction. Of course, they live in the wild and not in the lab, but basically we can use them to understand more about extinction. But to do that, we have to first define what the populations are, how small they are, how big they are, how connected they are, right? And then we can ask more specific questions about extinction in these small and isolated populations. And of course, we can then go back to thinking about the real world and what are the challenges that tigers face in the real world today. So here's it's a slightly outdated map, but it shows you the historic range of tigers in mustard. So you can see the tigers are very wide ranging. We're not talking about some finicky species. We're talking about a generalist species that really occupied, from cold environments like the Russian Far East, hunting in snow, to tropical rainforests in Southeast Asia. So this is a species which really lived in a wide variety of habitats and did fine, right? But given the loss of range, the loss of habitat, tigers have, we might expect, also declined in numbers, right? They've lost about 95% of their range. They've also seen extinctions already: the Caspian tiger, the Bali tiger, the Javan tiger, and the South China tiger, as recently as 1990. So the first thing which we did, well, not first thing, this is a recent study, but we did something, we tried to do this many years ago, was to actually look at a range-wide genetic variation of tigers using genomes to ask which populations of tigers actually have high genetic variation or not, and what can we understand about them. So this study was something which I'd been wanting to do for a very long time. I was lucky to go on a sabbatical on a Fulbright fellowship, where I was able to work with zoos in the US, which had, because I had no access to tigers from Sumatra, Russian Far East, and so on, right? So in order to get access to those individuals, in the US, many of these people who've studied them from the wild or wild caught individuals and their offspring in zoos, it was possible to, and of course with many, many collaborators, I think Anuradha is here, we were able to actually look at, variation across range, right? And we were able to then basically first interrogate the data and say, if we don't have any a priori expectations, what are the genetic units that pop out as populations? The lower two panels, a PCA plot here, and a structure plot show you this. So basically you can see here in the PCA plot, all individuals which are geographically associated with East Siberia are genetically close to each other. This is the Russian Far East or the Amur tigers, right? And similarly here, the Bengal tigers are all clustering together. The Malayan tigers are together and the Sumatran tigers are together. And you can also see in this structure plot, basically each line is an individual tiger, and their ancestry, the genetic ancestry is the color. So you can see that, all of these guys are homogeneous. They're one group. What's very interesting is, there's a few tigers, 3 individuals in the South Asia pool, which seem to share some ancestry with both Malayan and East Siberia. And these are these 3 individuals over here. I hope you can see my cursor, from the Northeast. Unfortunately, we did not have samples from Thailand. It's been very interesting to see the gradation of genetic variation from the Northeast going into Southeast Asia and island Southeast Asia. But also very interestingly, I'm going to, minimize this. The nucleotide diversity, or the proportion of genetic variation, by not a large margin, was the highest in South Asian tigers. So Indian tigers do seem to have a large proportion of genetic variation. And it turns out that Sumatran tigers actually have the lowest variation. But what's also very interesting is, you can look at, this could just be because our sample sizes are different, right? So if you actually do a rarefaction analysis to look at mean number of alleles per locus, and you do a rarefaction to see how this changes with sample size, and you compare these 4 populations, Amur tigers clearly have really low variation. And the reason it's going down is because of error. But basically, as you go with the Bengal tigers, we still haven't even reached a plateau. So we still, even with 20 individuals, we're still discovering variation. So there's a lot of variation in Bengal tigers in its entirety. And basically they have the highest amount of variation. But what was also really interesting is, when you look at these runs of homozygosity, now runs of homozygosity are a way to measure inbreeding. You can just look for a minute at this pedigree. So imagine that my parents are related, and that means they could get the same stretch of DNA from their ancestor, which was common, right? So the common ancestor which they had could give a stretch of DNA to both of them. And that same stretch of DNA comes back to me if my parents are related. And the longer that stretch is, the more recent is the inbreeding. So this is called a run of homozygosity, and it's been used in many species to quantify inbreeding. So like I said, the longer the stretch, so 2 MB is a long stretch, and the shorter the stretch, 100 KB, for example, is a short stretch, the shorter stretches are older. So basically you can see that all tigers show relatively short stretches. This is indicative of the older kind of bottlenecks or the declines that they've all witnessed, all tigers have witnessed. But what's very interesting is, when you start looking at the longer stretches, 1 MB and longer, it is the Bengal tigers, the pink guys, I love that it's pink, are popping up. This, by the way, is an inbred zoo tiger. Okay, so what that's telling you is that while together Indian tigers have a lot of genetic variation, particular individuals in India do seem to be inbred. So this was a hint we got, and so we were very curious to understand more about inbreeding. Of course, we know that tigers have been hunted, and so those shorter stretches we see could be because of these bottlenecks. There's a picture, for example, of a hunt from the British Raj times, and ongoing trade and continued poaching, illegal hunting of tigers. Of course, tigers have also lost habitat. So this is an extrapolation from a database called Anthrome, and you can see forest cover as it changes in India from 1700, 1800, 1900, and 2000. So clearly, if at all tigers were tied in any way associated with these green patches, you could imagine that their populations would be isolated. So both of these factors, small population size and isolation, which we identified earlier on theoretically as being important in pathways to extinction, are something which we are seeing, Indian tigers kind of move along those directions. So basically, we study tigers through a genetic lens, and we ask two types of questions. One is basically an ecological question. Are tiger population sizes small? Are tiger populations connected? This is an ecological question in a way, right? But we're also interested in purely genetic questions. What are processes like inbreeding or drift, which tend to occur prior to extinction, and can we detect these? So this is basically, mostly we do what's called molecular scatology. We walk in forests, we sample fecal material, sometimes other things. This has been changing over the years. We bring it back to the lab, extract DNA, and use markers to identify individuals, species, and so on and so forth. So here's the lay of the land again. The black polygons are the tiger reserves in India, and you can see that they are quite spread out and sometimes far away from each other. While there are 3,500 tigers, or whatever we can argue about those numbers, maybe 3,000, the median population size is 40 or 50 individuals. That is, most populations, most of these protected areas have about 40 or 50 tigers, and only 5 populations have more than 150 individuals. So for there to be low chances of extinction, we must have connectivity. This we know without any study at all, right? From what we know of other species, in order to kind of avoid extinction, we must have connectivity. So can we actually estimate population size itself from genetics? I mean, there's very robust ways of doing it through camera traps, but it was some work we did very, very many years ago, my first student, to try and understand whether you can actually count tigers with mark recapture methods. You walk through the forest, collect fecal samples, bring them back to the lab, identify individuals, put them in this statistical framework of capture and recapture. And basically, our estimates of tiger numbers were pretty much identical to the camera trap estimates in that year. The next questions that we've asked are about what the landscapes or connected populations of tigers are. So we identified early on that Runthambore appears to be an isolated population, and I'll talk more about Runthambore, but also that this is a large landscape which I don't think the population structure of it is very well resolved, but overall central India does seem to have high variation and more connectivity. And I'll talk about that in a little bit as well. I just realized that I don't have that much time, so I'm trying to go a bit faster. So I'm going to zoom through this because this is old work and I want to talk about new work, but we did some work on connectivity where we looked at what are the factors which allow for connectivity, if there is, in the Central Indian landscape, and others have done similar work as well. And what we found was that high traffic roads and high density human settlements provide resistance to tiger movement. And when we use this to try and understand basically future landscapes, we can predict if the landscape changes like this, what will happen to connectivity, and such approaches can actually be used to plan landscape development. In this particular case, our work was used in court to petition, amongst other studies, to petition for this wildlife underpass. So this is Kanha here and Pench on the other side, and this is a highway which is being expanded, NH7. But given that our work and others had found evidence for connectivity between Kanha and Pench, basically building this highway would disrupt that connectivity, disrupting a natural biological process. So the court found value in ordering the National Highway Authority of India to construct this overpass. Basically, wildlife can just walk under this. And this is a really big win for conservation based primarily on scientific data. So I'm going to now talk about these small and isolated populations, and I have two studies that I'd like to share with you. One is basically on inbreeding depression, and this is focused in Runthambore, and I'm going to walk you through it. So here's Runthambore. It's a spectacular park. It's in Rajasthan, and you can see in the background, there's a tigress here, and in the background is this hunting lodge, which is in pink, stone, as is common in Rajasthan. And tigers are actually really common in Runthambore. They're high density, you can see them easily. And so whenever we found these initial signatures of isolation, that signature was more because all the individuals in Runthambore are related. And so the whole set of individuals looks like one cluster, right? They're all similar, they're correlated with each other. So what we did was we sequenced genomes from across India, and we tried to look at inbreeding across between Runthambore, how inbred is, say, Runthambore, which is a small and isolated population, compared to two other populations, southern India, the Western Ghats basically, and some populations in central India, which we call larger and more connected populations. And what we found is, so you can see southern large connected is Western Ghats, central Western Ghats, and central large connected is basically Kanha, Pench, that landscape. So this is runs of homozygosity, and the proportion of the genome with runs of homozygosity larger than 1 MB. So the tiger genome is 2.4 MB. 2,400 million base pairs to 2.4 GB. So you go through the genome and you keep walking till you have a break in homozygosity. So how many of those stretches are longer than 1 MB, and what proportion of the genome do they occupy? So this is basically equivalent to an inbreeding coefficient. So for example, that SIB matings are inbred, right? So like that, we can have ROH also gives a measure of inbreeding. And what you can see is that overall individuals from Runthambore have a high FROH. A large proportion of their genome is in long stretches of homozygosity, whereas southern India is not as inbred, and central India certainly appears to be much less inbred, right? Now, not only are individuals inbred, but the stretches which are homozygous are also shared, okay? So this is basically, you could have many individuals who are inbred, but they're not from the same inbreeding line. So from a perspective of inbreeding depression, where you have two individuals who mate who are inbred, their offspring carry the same recessive deleterious alleles, and that leads to inbreeding depression. But if the inbred stretches are not shared, it doesn't matter. You could have inbred individuals and it's fine. But what you see here is that indeed individuals in Runthambore also share a large proportion of those inbred stretches. This is, for example, known to be inbred zoo individuals, and they share much more of these inbred stretches. We also wanted to then see whether inbred individuals have more deleterious mutations. I mean, inbreeding is fine, but does it matter, right? The way it matters is through deleterious mutations. So let me just walk you through this. So basically, we have a assembly, a tiger assembly. We have, RNA-seq data so that we can identify genes, right? And once we know the genes, we can use something called variant ensemble predictor to make a prediction if there is a SNP or a mutation in a specific gene, Might that lead to a loss of function or potentially a missense mutation? Missense mutations are called deleterious. Loss of function is basically when you have a stop codon somewhere in the middle or something like that, which you would imagine that there's absolutely no expression, there's no translation of that gene at all. It's predicted, it's theoretical, right? But what you see here is that if you look at the number of, and so this is damaging homozygotes, is all of them together. So clearly, if you would expect that inbred individuals have a higher mutation load, right? They have more recessive deleterious alleles, right? More deleterious alleles, whether recessive or dominant, whatever. So what you can see is that indeed, individuals who are more inbred, where FROH is higher, have more damaging mutations. They have more, deleterious plus loss of function mutations compared to individuals who are not as inbred. And this is irrespective of population. That is, irrespective of whether you look at the color of the dots, there is a positive correlation. So more inbred individuals also have more damaging homozygotes. So given that most recessives, most damaging alleles are homozygous, this means that there could be inbreeding depression in inbred individuals. Okay, I hope that's clear. The trend is not as clear with loss of function alleles. And the reason for that is as follows. When we compared the load or the number of loss of function alleles between individuals in the large connected and the small isolated population, what we found was really interesting. Actually, the small isolated population had fewer loss of function alleles than the large connected populations. This was not true for the slightly deleterious or the other missense mutations, right? Not all missense mutations may be bad. And by the way, you can't see it here, but this box here is neutral mutations, right? So what this is suggesting is called purging. So there's this idea in conservation biology, population genetics, you have a small population and you have inbreeding, you know that there are deleterious alleles. When individuals are inbred and they mate, if there's something we just say a lethal, then the individual will die. And this will remove or purge the very bad loss of function or deleterious mutations. So what we seem to be seeing is a signature of purging. And I'll show this to you in another way. So these are what's called site frequency spectra, or the frequency of the proportion of loci which are very low frequency, 0.1 to 0.2, 0.2 to 0.3, and so on. So the frequency distribution of loci, right? So what you see here is in the small and isolated population, you seem to have a jump, and this is neutral is blue and potentially damaging is orange. So wherever you see a difference between orange and blue, it seems like something may be going on. Okay. So you can see that actually in all cases, most strongly in all tigers, okay, all Indian tigers, that the proportion of loci in the 0 bin and the 1 bin is high. This is basically genetic drift, fixation of loci by chance because they're damaging, right? And this is basically a signature of purging. That is, those loci have potentially been removed. So I know this is all very theoretical, but the idea is pretty exciting that tigers have already experienced inbreeding depression, and that inbreeding depression has removed like potentially the very deleterious alleles. However, what's really interesting is the alleles which remain in Runthambore are at very high frequency, which means that Runthambore, the small isolated population, could still experience inbreeding depression. So the old-fashioned way of characterizing inbreeding is to look at pedigrees, and if we could combine this inference with actual individuals with phenotypes and understand their reproductive success, that would be really cool. So we're trying to do this. This is the purported pedigree of individuals in Runthambore. This is all the individuals. You can see there's a lot of cross mating and inbreeding and so on. And what we would also like to do is to understand these deleterious mutations better. So one possibility is to actually do a lot more bioinformatic curation, compare our mutations to mutational databases like polyphen and so on, known mutations, as well as known conditions in cats like a polycystic kidney disease, hip dysplasia, and cardiomyopathies. Cats are known to have these conditions, and see whether any of these mutations fall in those genes. They do actually. We do have mutations in those genes, even in the Runthambore tigers, and then try to understand or predict with some functional assays whether they might affect individual fitness in the wild. So going back to this pedigree, the way we are trying to put it together, this is now different, right? I can't put together a pedigree by just going and sampling fecal samples randomly on some road in the forest, because I don't know who that fecal sample belongs to. If I do that, I only get a representation of the population, but I want to know this individual's DNA. I want to capture this particular individual's DNA. So what we figured out is actually that hair is a pretty good source of DNA, shed hair. And especially in sites like Runthambore, you can watch tigers, and once they go away, you look around, you find lots of hair. You can bring that back to the lab, extract it, and actually just sequence it. And when we did this initially, we found that there's a lot of variability in shed hair. A large, sometimes very few reads from shed hair map to tiger, whereas other times a lot of reads map to tiger, right? But definitely shed hair was way better than fecal samples. Fecal samples have so much other DNA, right? Like fungal DNA, bacterial DNA, prey DNA, and so on. And the proportion of tiger DNA is very, very small. So in that sense, these shed hair samples are a good way to potentially identify or get genomes from individuals. And we've used this in like a toy example. We're still doing this. We've collected mothers, offspring, and potential fathers for about 4 years now in Runthambore. This is going to take a long time for us to put together this pedigree. But when we did look at some cases of known and unknown pedigrees, we were able to, in this case with mitochondrial DNA, resolve mitochondrial whole genomes, resolve which individual actually was the daughter of, this is like a famous matriline of Mushli in Runthambore, and which was kind of a very different individual. This individual looks to be almost like it's from some other population, maybe bringing in a new matriline, which was not known. So I'm going to talk to you very quickly, since I'm running out of time, about Simlipal Tiger Reserve, another isolated population. So what's fascinating about Simlipal Tiger Reserve is that they have these really beautiful pseudo-melanistic tigers. Why do I say pseudo-melanistic? Because they're not completely black, right? They're sort of black, like someone dropped some black paint on them. And it turns out that, there's been studies on this type of a coat pattern and a disruption of pattern by this guy called Chris Kalin and Greg Barsh. They work on cat coats and genetics of cat coats. And what they found was that mutations in this gene called transmembrane aminopeptidase Q, TACPEP, results in, like, for example, in the cheetah, in the disruption of the pattern from this rosettes to this, and in cats from a tabby pattern to kind of a blotched pattern. So of course, the minute we saw this tiger, we thought, it's not like we saw it first. People have been seeing these tigers for a long time, discussing them. A.K. Lala Singh has a wonderful book called Born to be Black, where he talks about this for many, many chapters. People have wondered about this, the mystery of the black tigers of Simlipal. And of course, it looked very easy in a way. It looked like it was probably a mutation in TACPEP. But how do we investigate that? In the wild, Simlipal has very low tiger densities. You don't see tigers at all. Luckily for us, in the Nandankanan Zoo, a litter was born which included 2 melanistic individuals, one white pseudo-melanistic individual and one black pseudo-melanistic individual. So we were able to then collect both saliva, blood, etc., do whole genomes, and do a candidate gene-based approach to look at the pedigree of these pseudo-melanistic tigers. And we identified a point mutation in the TACPEP gene, which results from a histidine converting to a tyrosine. This particular position is in the membrane binding domain in a highly conserved domain. So basically, we expect that it may render the protein non-functional. So now we found the mutation, but then was that what was occurring in Simlipal? This is what is there in the zoo. So we went back to Simlipal and over 2 years, walked the trails, collected fecal samples, and we sampled about, I don't remember now, about a 70 or 80 scats, came back, extracted DNA, identified tigers, genotyped individuals. We were able to totally genotype 12 individuals. And then we looked for the presence or absence of this mutation. And we could either get a homozygous individual, a heterozygous, or a wild type individual. And basically what we found, this is the frequency of the different mutations and wild type allele we found. And it turned out then that the frequency of the mutation is almost 0.58. This is really high, right? Because these individuals are not seen anywhere outside of Simlipal. Really? So maybe we just miss them because they're present in a heterozygous state, because heterozygous individuals look wild type, right? So together with a lot of collaborators, and thanks also, big thanks to Anuradha and CCMB team here, we were able to look at almost 400 individual tigers from across tiger range, mainly from India, and basically we did not find the pseudomelanistic allele mutation anywhere other than in Simlipal and in two zoos. One is in the in the Chennai Zoo and in the Nandankanan Zoo. Okay, so this was crazy that the mutation seems to be absent or very rare everywhere except in Simlipal. So basically that implicates drift, right? That somehow in Simlipal, this mutation has reached high frequencies by chance. An alternative hypothesis is selection, and I'll come to that in a bit. But when we looked at population structure or how differentiated is Simlipal from other populations, we saw that it is indeed, quote unquote, genetically isolated. These are other populations in central India, Tadoba, Satpura, Kanha, etc., etc. Also, when we looked at relatedness between individuals in Simlipal, this is with very few markers. We only have about 85 SNPs. We only have fecal samples from Simlipal. We do not have genomes, unlike Runthambore. We do that, we see that relatedness between individuals is quite high, almost as similar to Runthambore, which we know is an inbred population. So Simlipal also appears to have inbred individuals. Now, why is Simlipal isolated? So we did some landscape analysis to try and look at this. So basically, when you allow dispersal, so basically this is the landscape, you can see immediately that there is no other protected area where there are tigers very close to Simlipal. Okay, so Simlipal is just very, very far. Okay, so if you set, depending on this landscape and what's there, and allowing tigers to move, if you set the dispersal threshold to 200 kilometers, that is, they move about 200 kilometers, there is no way any tiger would reach Simlipal. On the other hand, when you have a dispersal threshold of 500 kilometers, there's a low chance that you would make it there. Only with very high dispersal thresholds, like 1,000 kilometers, is the chance of Simlipal being colonized pretty high, right, from somewhere else. So what this means is that Simlipal is isolated, not necessarily because of fragmentation, but simply because it is very far from any other source of tigers. And those tigers, if they did come in, would dilute the frequency of TACPEP and not, it would not go as high. And finally, we did some simulations to basically look at, what kinds of bottlenecks, what kinds of scenarios could result in such a high frequency of TACPEP mutation in Simlipal. And basically only when you have a very strong bottleneck of say 2 or 3 individuals, is there even an appreciable probability, a small chance that you would get such a high frequency inside Simlipal compared to outside. So I've talked to you a lot about tigers and their recovery and the importance of connectivity. I've highlighted how, we cannot have isolated populations and so on. But when tigers come out, what happens is there's conflict. And this is a very exciting project we are doing. I hope we finish it at some point. It's been going on for a long time. I'm almost done, so don't worry. But what we're doing is we're actually trying to sample cattle kill to understand which tigers are actually involved in conflict. And this is a camera trap video. These are two cubs. Yes, they're cubs. You might be surprised, but cubs are pretty big. And that's a cattle kill on the side. And you can see the tigers are basically, they're eating. So we can set up these cameras near a kill site to actually see, who's coming to the kill. But that's kind of standard methodology. See, this individual is really curious about the camera. So he's kind of going to come and sniff it. Yeah. So fun things you get to do in this research. But what you find is that when you look at these cattle, there are bite marks and there's like a lot of other signs, hair, where the animals have been eating, there's poop around and so on. So we treat each kill site almost like a forensic investigation where we can actually sample all these different kinds of things, jugular, bite marks, lick marks. This is a lick mark here. You can see the skin is all flattened. You get saliva from there. Fecal samples and so on. And this, we can then use this to identify individuals. This is an ongoing project, like I said. We can then design this framework to say, okay, how many samples can we identify to individual level? Who is the true predator? Is it the true predator? Is definitely the one which did the jugular bite. Are there other individuals which come and are associated with the kill site and so on and so forth? So understanding which individuals are involved in conflict will allow us to potentially suggest strategies for mitigating or reducing conflict. And this is a project funded by National Geographic. And I'm really lucky to have, an old friend and colleague, Prasanjit Yadav, who's a photographer, also involved. And this is my student, Himanshu. We also do some work on bats in a bat harvest in in Nagaland. And this is something which we've been working on for many years prior to COVID. And something which I think will be important part of our research program, I hope, going forward. So I just have to acknowledge that I just give talks and write grants and papers, and a lot of the hard work is really done by fantastic people I have the real good fortune to work with, and a lot of people who invest in our work. It's really expensive. Couple field work with genomics. And of course, permissions and so on and so forth. So I will stop here and I'm happy to take questions. Sorry, I hope I wasn't like, I just zoomed through.
I think it was very nice. Uma, can you see the chat box? There's one question.
Yeah. So there's a question from Meher. Did anthropogenic factors play a role in losing connectivity with Simlipal? As in tigers' historical distribution, there aren't any gaps because then the mutation could also be a recent one. So yeah, so we don't know how recent the mutation is. To look at that, though there's always a lot of error associated with those estimates, we would have to sequence around the mutation and look at LD, linkage disequilibrium. And for that, we need a good sample like blood or something like that, tissue or something, which we don't have any from Simlipal. So we've not been able to look at that yet. And also I said I was going to talk about selection. That is also how we would look for selection. We'd have to look at other loci and see how differentiated they are compared to this one. Is this at a higher frequency than you would expect by chance? With the limited number of loci we have, that's not the case, but 85 loci is not enough really to answer that question. So see, the thing is that there are other tiger populations close by. So there's a population called Satkoshia. There are tigers in the Sundarbans, but the Sundarbans, while close as the crow flies, is across Calcutta and a lot of barriers, right? And of course, a very different habitat. But potentially, if you see Simlipal tigers do have Central Indian ancestry. So they're probably part of the larger Central Indian landscape. And there probably were a lot of tigers in Jharkhand and Chhattisgarh, where today there is almost nothing. So there was probably historical connectivity. What connectivity was there to the Tarai? I'm not sure. And that's something which I think we still don't understand very well, that nexus of, Eastern, Central India, Northeast and Tarai. But yeah, I think that right now the problem is really that there's really no source population which is close by. So even if when we look at Central India and we say it's a set of connected populations, all the population numbers are very small, 50, 100 and all. No. So it's a meta population. The only way such populations are sustained is by recolonization many times. Okay, if there's extinction, there's recolonization. They're not long-term stable populations. So in that sense, all of our populations need to be able to be colonized again. That is not the case for Simlipan. Okay, Saket, this is a question which is something which keeps me up at night. Is it feasible to increase genetic diversity by artificially breeding individuals from different populations? Of course, if the only goal is to increase genetic variation. But I worry because, first of all, I'm talking about these putative deleterious alleles. I don't know. Missense mutations may not all be bad. They may even have sometimes a positive effect on fitness, right? We don't know. We don't know enough. So that's something which I'm hoping to explore in the next 5 years, if I am able to secure funding for it, is to actually prioritize mutations based on which ones I think would definitely have an effect on fitness, look at their functional effects maybe, and then maybe rank populations as more high risk or whatever based on their mutations, more at risk of inbreeding depression, less at risk of inbreeding depression. Of course, it's very ambitious and it may not happen. On the other hand, without a doubt, we must strive to maintain structural function connectivity. We cannot give up on that. We cannot say, oh my God, it's too tough. Let's just fence the tigers. Let's just airdrop them. Let's just manage the whole thing. Then it's a zoo. Okay. I mean, I guess we can do that if we have no choice, but I think that we should always try to maintain functional structural connectivity. And how do we do this with the face of conflict, in the face of increasing human population density in India and increasing demands of development? Well, there are other targets of the government like restoration. We've all signed on to this bond challenge where we've said we're going to restore a lot of habitats in India. Well, maybe these restoration can coincide with connectivity, or we can use different, or climate mitigation. We have to do afforestation or habitat something for climate mitigation that can also help tiger connectivity, right? We have to think of what's it called, triage. We have to think of ways to get multiple benefits from similar actions on the ground, because biodiversity loss and extinction is really not an option. Yes, maybe ultimately, so maybe we will have to introduce individuals, but the success of individuals is not only based on their genetics, it is based on their behavioral integration into the population. Is there enough prey for those individuals? So ecology and behavior also play a strong role in whether those individuals will survive. So I think we have to have a very integrative approach to think about. Yeah. Is it always conflict when tigers share a kill? Oh, no. So the conflict is not about the tigers sharing the kill. The conflict is about the tigers killing cattle which belong to villagers, right? So basically when tigers kill cattle, the person whose cattle it is, loses a huge amount of money and they get compensation, but it's not the same, right? You lose, it's like maybe, you lose something, it's a loss. And okay, I didn't show these results to you, but we just did a meta-barcoding study on diet, and 40% of diet of Runthambore tigers, it's a protected area inside the park, is cattle. So what is wild now? Who is wild? Tiger eating 40% cattle. It's not killing a wild prey and eating. It's not what we think of as a tiger, right? I mean, we don't think of it coming and easily eating cattle. So this is a big thing. And I think in this case, it could also be influenced by changes in policy. after certain policies of cattle killing, there are tons of cattle roaming around, abandoned. So from a tiger's perspective, this is very good prey. There's tons of them just roaming around, and they're not even probably as agile. But what we do need to understand is that it's going to be a problem that's here to stay. So we have to think of how to manage it. I can always think of tech solutions. I naturally veer to tech. Tech is not the solution in conservation problems. I have to say that. But for example, what people are now doing is they're using nanopore on field to sequence what is that individual. Because if you're going to say, send it to the lab, next Illumina run, it'll take months. That is useless to conservation. Today, some cattle has been killed. Tomorrow, another cattle. Someone has attacked a human. We need to know now who is that individual, right? So sometimes the speed is also very important to find out about conflicting individuals. So there, I think maybe some new technological approaches can be applied, but it's very important to link a genetic ID to an individual. Like I can go and sample whatever DNA I want, but I don't know which individual to catch. On the ground, I have to catch a tiger. I can't look at it and tell its genotype. I'll need to sample it, I have to catch another one and release it. So we need to also build databases of individuals, like a biobank, so that we know, okay, this is the individual, this is the genotype. When I go to a conflict site, I pick up this genotype, it's this individual, and this is the problem individual, something like that. But it has to be very dynamic, otherwise it will be useless. At this point, it's just a research question. It's not fast enough to be of use in management.
Okay, well, there is also a question in QA by Ashish Bihani.
I see. Yeah. So is there any initiative possibility on doing animal-focused archaeological investigation that used to have these populations but may have lost them now? I think this is a great, option if we can actually investigate the past. So we have just started some work with skins because I'm curious to know about this purging, whether there were deleterious alleles in the past which were lost. But I think your question, Ashish, is more about, are you thinking about tigers which may have existed earlier close to Simlipal or something like that? So I guess, we can definitely look at skins, but we don't, I guess we can also, okay, so there are very few places in India where tigers were not there. So almost everywhere they were there historically, right? So in that sense, it's a very recent, like last 100 years that they have gone extinct. Even in Gujarat, there were tigers, right? But they have gone extinct there now, right? So I think it's something which maybe historical analysis might shed some light on. But yeah, so for example, it would be nice to know that from the skins from areas like Gujarat, where there are no tigers now, did you lose something in particular or not, and so on. Yeah. So Umapati, hi Umapati, asks, do you think metapopulations exist between central, south, and eastern populations? No. I think that south is disconnected. Eastern Ghats, I don't know. I'm not sure about NSTR. So I don't know. Actually, I should say I don't know. I don't think so. But maybe there are still, even though you say population structure, we've been studying tiger population structure for 10 years. We still don't know a lot of things about specific populations. We don't know about, say, this Northeast, Central India, Tarai connection, and so on. But yeah. So also he asks, if not, is it possible to establish near future as tigers are learning to move in human dominant landscapes? So yes, anything is possible, but what we need to try and think about is, as we move towards assisted migration or movement of individuals, that's what's going to be the, like I stomped my fist and said we have to maintain connectivity, but I'm stomping my fist in my house, doesn't matter. But we will have to move towards assisted gene flow, moving individuals to reduce inbreeding or whatever. So I think at that point, we should plan it. We should plan these rescue strategies. And I still feel like we can talk about them theoretically, but I would like to do some more work before, really making that suggestion. But I think we should focus more on deleterious alleles. That's what I feel. Negative impacts, yeah. Is there a central data repository in India for tiger population related data? So it'll be wonderful. As you know, we are very bad at repositories. So all my data is all my data is on GenBank, at least in terms of the genetic data. But I was thinking about it, maybe making a website or something where people can actually, because of course you can download the data from GenBank, but then there's very little metadata associated with the GenBank, the SRA IDs, right? So maybe a more of an interactive database where we could, talk about those genomes, what specific thing to, also like a learning tool. You can say, okay, this is how you would, these are the runs of homozygosity, these are the deleterious mutations. People can actually explore those things a little bit better. And as we have more and more genomes, maybe that's something we should really think about. On the other hand, I'd be very excited once we have this data on pedigrees and all in Runthambore, and also more data on individuals, to create a more public facing kind of something like about individuals, right? Like this individual is related to that individual. And I think that's a really exciting, again, outreach tool. But I think it would require some design. And the other thing which I'm very fascinated with, I have not done anything about, is there's tons of tiger pictures on social media, right? And we could actually pull all those pictures and use AI probably to identify individuals and include the different pictures of individuals together, and maybe understand something about phenotype or morphomes, right? And then look at relationships between individuals, what is inherited, what is not. So there's a lot of potential for, because a lot of photographs of tigers. Sorry. Some tigers, there are thousands of pictures, thousands of the same individual. Sorry. Sorry, I get a little carried away. So yeah, is there some other question?
No, definitely. I think that's a great idea. It's also a way to get people involved in the conservation, right? And I guess from what you're saying, some tigers are social media stars, so we use them. But yeah, would like to talk to you more on that. If you don't even have the initial database or data, then that would be the start.
Yeah. And there's also a lot of camera trap images, right?
Yeah.
Those are not shared publicly though.
True.
There should be a database of those also. I mean, Government of India has been doing camera trapping across India to count tiger numbers for 10 years. So all those data also should be somehow available or usable or something like that. But yeah, I think that this idea of databases, like for example, even for zoo individuals, because one option might be, someone talked about genetic supplementation, that we actually have to work with zoo individuals, because many times reintroductions fail. And then if it's a tiger which you got after 3 years of requesting MP Forest Department, and then you brought it and it dies or it's killed, it's like too much of negative backlash, right? So zoo individuals are a little less, they're a bit more dispensable maybe. But yeah, so I think that these databases would be really nice. And I remember Eric Lander visited us once and he said, you have only 3,000, 4,000 tigers, sequence them all. So if only we could get all of them.
Why not?
From a human perspective, it looks very, very few numbers. So yeah, great question. Yeah.
Thanks.
looks like there aren't any more questions. Any participants want to ask questions live? Okay. Hey, thanks, Uma, for the wonderful talk.
No problem.
I'm sure all of
Thanks
you.
for the invitation. Yeah.
Oh, there's one more
Great.
question. Do you want
there's
to
one
take
more
that?
I don't see. Oh, yeah. So as an aside to Ashish's question, do we have any fossil samples? No, not that I know of. The only fossils are from South China. But yeah, so I have a feeling that in general, tigers are quite recent. So like, even if you look at the divergence between the subspecies, it's like very recent, like 20,000 years. So I don't know, maybe there certainly aren't, at least I don't know of any fossils, in India. If they were, they would be too old. Like basically we are interested in the last 1,000 years, that's what we're most interested in. And we have, skins from up to 150 or so years ago. In the London Museum, actually, Janvi, there are skins from Afghanistan, where tigers are now extinct from about 150 years ago. But the thing about working with skins now is the methods have changed. Now it's much better with these capture probes and stuff. You can get a lot more data. So we're hoping to actually look at them a little bit more. Okay, thanks everyone, and hopefully we'll.
Thank you so much for a wonderful talk.
Yeah.
Yes.
Okay. Bye.
Yeah.